Now let's start with the fluorescence experiment. In the UV visible absorption spectroscopy, we have noted down the absorption maxima of various proteins. Now we are interested in finding the emission spectra of those proteins, how it looks actually. So for that, what we need to do, we need a fluorometer. So what is a fluorometer? This is a fluorometer. So fluorometer is basically an instrument which is utilized for measuring the fluorescence spectra of a protein or any compounds. So this fluorometer has a compartment like the UV one and a screen where we can completely monitor our UV spectrum. Now compared to our UV spectra, this fluorometer uses only one cuvette, that is only one cuvette. Here this cuvette is a quartz cuvette and it is much more costlier than the UV cuvette. And one of the important thing to note down in this cuvette is that all the four sides of this cuvette, here we can see, are transparent. There is no frost side out here. This is because, as we have seen in the theory classes, in fluorescence spectroscopy, the light is transmitted in the right angle direction. Once it goes out here, it is light transmitted in right angle direction, the fluorescence light. So that's why here we have taken the cuvette and all the sides are equally transparent. Now this cuvette should be cleaned, completely cleaned. It's better to clean this cuvette with acetone and dry it completely before using it. Now we have washed it with water for the time being. Now let's start with our experiment. Now we are filling this cuvette initially with double distilled water and taking a spectra first of all. Now we have taken this cuvette and filled it with 3 ml of double distilled water. Now we will clean all the sides properly. One important thing to note down here is that please don't touch any of the sides of this cuvette. You can touch here in this top or you can touch those corners out here, but don't try not to touch this all those 4 sides. You can touch the top layer where the solution is not there. Okay. See whether if bubbles are here or not. We can see a few bubbles. Just touch it with the pipette. Basically this is not any protein solution, this is pure water. Now we have taken double distilled water in the fluorescence cuvette and we will measure the fluorescence for double distilled water for the time being. Now this is the fluorimeter instrument, this one is the lead of the instrument. We will open this lid and here we can see a compartment. Now in this compartment, interesting thing note down here is that only one holder is present, unlike the UV instrument where 2 holders are present for reference and for sample, and that's why we have to repeat this for 2 times. Now this one is for the reference, we are using this solvent water. Now we are placing it in this holder. However, here we can see one is the entrance slit. Near the entrance light, excitation light will come. Here the monochromator is there, excitation monochromator just behind the slit, and it will go through this sample and the fluorescence emission will be detected at right angle to it. So another slit out here, exit slit is present here, where it will detect the fluorescence emission range, which we will place in the method part, and the exit, in the exit part, the emission monochromator will kind of scan the emission range. Now we are placing this cuvette gently out here in the holder, and here one thing we can see here, the 2 pipes are there. So a curious mind may ask, what is the reason for these 2 pipes? Basically it's for other experiment in which we are doing any temperature dependent study. Here these are the water inlet and outlet pipe. Water comes from one place and goes out from the other, basically it heats up the sample. We are not interested kindly in this one, we are interested in basic spectroscopic techniques. So after we have kept this solution in this holder, we will just place down this lid. So this is the software for this instrument where we are doing the fluorescence spectroscopy analysis. Now different softwares are there for different types of instrument. For this instrument, this is FL solution is the software. Now we want to go to the method. Initial part we have to set, we have to give information to the software how we want to proceed with our experiment. For this we have to go to the method part. In the method part we want to do a wavelength scan. Other parts are there like 3D scan, 3D time scan, but currently we are doing wavelength scan. In the instrument part, we are interested in emission. Other things we can do like excitation or synchronous, but currently we are doing emission And the data mode is fluorescence, not luminescence or phosphorescence for the time being, but fluorescence emission we are interested in. The excitation wavelength out here is 278 nanometer. The emission wavelength is a range of wavelength where it will start from here. The emission wavelength start is around 295 nanometer. Generally, it is kept around 10 to 11 nanometers higher than that of the excitation wavelength. The emission wavelength will end at around 400 nanometers. So, I have to give a band, a series of band in which the emission will take place. Now, the scan speed is set to around 240. We can vary according to our requirements. The delay is 3 nanosecond. Now, the slit width. Here the exit and the entrance slit is basically the excitation slit is the emission slit. It is on 10 nanometer, it is fixed. However, it depends upon instrument to instrument. For instrument with much more higher resolution or much more advanced fluorescence spectrometer instrument, the slit width is basically kept at around 2 to 5, and the slit will varies depending upon the type of fluorophore, the type of component out here in the instrument or out here for analysis, and also upon the sensitivity of this instrument. Fluorescence slit width for excitation is around 10 nanometer, and here its emission is also 10 nanometer, and other things are kept as default. Now, we are clicking OK. The machine will just take a few seconds to initialize upon itself. Now, we will give a pre-scan. This is a requirement for this particular software and machine, and the pre-scan part, what the machine basically initializes itself depending upon the instruction it is fed in the method region. After pre-scan, what we need to do, we need to go to measure. Now, once we click measure, here the scanning has started, and we can see a type of spectra, a non-uniform spectra basically appears, because we are taking the spectra for double distilled water which has no fluorophore present in it, just a background spectra for this one. Background spectra is required, that is the solvents of the spectra are required in which the protein or the fluorophore is dissolved. Now, we are done with it. Now, we will proceed to the next observation. Now, we will load protein solution and see its spectra. Now, we will see the fluorescence emission for a protein having tyrosine residues only, that is RNase A. We will open this lead, take out this double distilled water basically. Now, what we need to do is that we now need to pipette out around 10 microliter of double distilled water. Out here we are taking a 10 microliter pipette. So, we are taking 10 microliter double distilled water, and now we will be taking this RNase solution. We are taking 10 microliter of RNase solution and slowly adding this in the cuvette and mixing it properly. Generally, the amount of protein, or rather the concentration of protein used for fluorescence should be much lower than that of the UV instrument, because fluorescents are much more sensitive than that of UV. So, now we will again place this one in the compartment. One easy method is that you do not have to basically consider which side to face this or which side to face this one. You can keep it in any side, any way you like, unlike the UV part. We will close it. So, now we have taken RNase solution and let us see its fluorescence. Initially, a pre-scan required. After pre-scan, let us see the measurement part. Here we can see, here this is the emission max, lambda max for emission of this tyrosine residue. Now, this is the fluorescence spectra for RNase A. Now we can actually see it like this way. Increase it here. We can see the fluorescence spectra basically for RNAs is something like this way. This one is for the baseline, or not baseline, this one is for actually the solvent, and this is the fluorescence spectra. Maximum around comes value is around here. We can see the maximum value, it's around 304 to 305. So this is the fluorescence spectra for RNase A, which is for tyrosine residue, and no tryptophan is present. Now we have seen the fluorescence emission for RNase A protein, which contains tyrosine residues. The RNase A protein having tyrosine residues gives a fluorescence emission of around 304 nanometer. Now we are shifting to another protein, that is HSA, whose lambda max we have checked from UV. This HSA has tryptophan residues in it. Now we have taken the cuvette containing RNase A solution, cleaned it completely with double distilled water, and then we have taken this solution of HSA in it. Now putting this solution in the fluorescence chamber out here, closing this lid gently. Now let us see the spectra, how it comes. Now we have made a analysis in the measurement. Here we can see a plateau type appears out here. So basically what is this? This is not the correct analysis. Basically here the problem, mainly we might estimate that saturation has taken place. The intensity of the lamp is not sufficient enough to make this analysis. So what we need to do in this case, once you find a plateau, not only for fluorescence and also for UV1, you need to dilute this solution. So we have to take the solution out and dilute it. Now we have taken the solution once again. We will do a pre-scan. After which we will measure. Now here we can see the plateau is no longer there, or rather a spectrum has taken place like this way. So we have diluted it by 50%. Now this is the emission spectra. Here we can see this is the actual emission spectra for our HSA. Now one important thing to note down here is that the lambda max here we can see comes generally around 300, just over 340 to 345 or 347, it ranges upon different proteins, and for HSA it's around 344 or 45 like this. Okay. So now the next protein we will consider is lysozyme. Now we have seen the emission spectra for RNase A containing tyrosine and HSA containing tryptophan. Now we are seeing the emission spectra for lysozyme, which are both tyrosine and both tryptophan. Now interestingly, we will see how the emission spectra changes when we change the excitation wavelength. Now this is the solution for lysozyme. We are taking it in this lid out here. So we have seen emission spectra for tyrosine excitation of around 278 nanometer, expected emission spectra to give an emission maxima at around 304 to 305 nanometer, whereas we have obtained a different emission spectra with a maxima of around 340 to 345 nanometer. Now this emission spectra basically resembles to that of tryptophan residue. Why this is the case? Basically in absorbance spectra we have seen for lysozyme it gives a broad absorbance spectra. So tryptophan basically have absorbance around 280, and it might the excitation is although around 295, but tryptophan is absorbed around 280 nanometer. So once we are exciting the sample around 270 nanometer, apart from tyrosine residues, the tryptophan residues are also being excited. Now why can't we see the tyrosine residues, that the emission from tyrosine residues, although it is getting excited? The reason is simple. As we have studied in the theoretical classes, basically what happens, the quantum yield plays an important role out here. The quantum yield for tryptophan is much higher than that of tyrosine residues. So although tyrosine residues are giving a fluorescence emission spectra, but out here it is completely being overshadowed by tryptophan residues. So we can see here, once the protein contains the tyrosine residues only, we can deal with tyrosine emission, but once the protein contains both tyrosine as well as tryptophan, we should always consider with tryptophan emission. So in conclusion, what we can say from today's experiment, we have seen the UV visible absorbance spectra of simple amino acid residues as well as the protein composing of different amino acid. We have seen the RNase UV absorbance containing only tyrosine residues. We have seen for HSA containing tryptophan and lysozyme containing both tyrosine and tryptophan. From UV absorbance, we can measure the concentration as well as we can know the excitation wavelength for fluorescence from the lambda max value. Now coming to fluorescence, what we can see, the fluorescence emission, we have to set the excitation wavelength at a particular value and emission range. We generally start from around a value which is 10 to 12 nanometer ahead of excitation. After that, we have seen the emission for tyrosine fluorescence from RNase protein, for HSA the tryptophan fluorescence, and the most important factor for lysozyme what we can see is that for protein containing both tyrosine and tryptophan, we should stick to tryptophan fluorescence. This will be utilized in studying the protein ligand complexes and also protein denaturation studies. So fluorescence method is a widely used technique for studying protein complexes and also protein structure. Thank you. I hope you had a great time in this course. So we are going to see how to measure the absorbance of a sample, a protein sample, using a nanodrop UV instrument. We have seen how to measure the absorbance using conventional UV spectrophotometer. However, more advanced spectrophotometer here is a nanodrop. So what is the importance of nanodrop? The importance of nanodrop lies in the fact that we can use very small amount of sample. Previously we have seen that we have to use a cuvette out there, and cuvette generally requires around 600 to around 1 ml of protein sample, protein solution, but here we can use around 1 drop, and which is equivalent to 1 to 2 microliter and not at all milliliter. So we just need a pipette here and around 2 microliter pipette, and we can add here and we can measure the absorbance. So let's get familiar with this new advanced nanodrop UV instrument. This instrument basically has two major components out here. One is the screen where we can see the values. Once we operate it, we can come across this part. And the second part is this basement. Here in this basement, we can see two components out here once again. This one curved part, and once we lift up this curved part, we can see there are again two spaces out here. So this curved part is generally known as the handle of this instrument, and this part is known as the pedestal. So what basically goes out here is that we add this nano drop, or rather a small 1 to 2 drop of the sample out here in this part of the instrument. This is not the pedestal, and then we actually lower down this handle and the machine takes its absorbance. In addition, there is also a facility for adding a cuvette out here. Here there is a small compartment which is currently shielded because we are using it for nanodrop only. So here we can also add a cuvette. So this instrument serves two purposes, one for this nanodrop and the second is also for cuvette, where we can use around 200 to 300 microliter of sample. So let's start with this instrument. For that we have to first turn on the main switch, and then there is a body switch just behind this instrument which will currently operate. So we just switched on this instrument. So we switched on this instrument, and we can see here there is a light has started glowing, and the instrument is undergoing initialization like other previous UV instruments, so it will take some time for initialization. And in between initialization, please keep in mind, as we have previously seen, we should not take on the lead of the UV instrument. So similarly, we should not open this handle while the machine is initializing. So instrument is initializing and an option is given. So we should not leave this on. Here we can see in the option, do not leave on during initialization. About the card. আচ্ছা
দারুণ পেয়েছে আমরা ওই রিড ফিট কোরিলেট করে না আমরা তুলবি না দারুণ একটা ল্যাপ করেছি
আচ্ছা
কনভেনশনালের সাথে এটা
আচ্ছা যেটা বলছি এবার সেখানে আমার যেটা ইনিশিয়ালাইজ হয়ে গেলে আমাকে একবার একটু বলে দিলাম যে স্নিগ্ধা বলে দিয়েছে
ডিএনএ করবো না
So the machine is initialized and this is the home screen of this instrument. Here we can see various types of options starting from nucleic acid, proteins and other things. In a nucleic acid part we can see that we can measure the absorbance of double-stranded DNA, basis that is single-stranded DNA, RNA and other different types of DNA for DNA experiments. Now coming to protein part, here we can see we can measure the absorbance of protein at 280, then labeled protein if we need. And some proteins which do not have those tryptophan or tyrosine, here we can measure the protein absorbance at 205. Apart from this, we can also do BCA assay and other Bradford, Lowry, etc. things. And in addition, here we can also use this OD600 part. In this OD600 part, we are measuring the bacteria culture. And one thing to remember is that we should use cuvette for this and not the nanodrop. It's preferable to use a cuvette for OD600. For this current experiment, we will be concentrating on protein. And however, in this protein, here we are using this protein 280 because the protein of our concern has tyrosine residues. Now, once we have clicked this protein part, here we will go to this option and the settings option will show this auto naming and use cuvette. And if we use cuvette, we will tick this option. Here we are showing this path length and everything. However, for this nano UV experiment, we are not using cuvette, so we won't tick it. And simply, without ticking it, without checking these boxes, we will press done. Now, we will go to this type select. Once we click this one, we can see the drop-down menu. It's showing BSA and lysozyme and other different types of proteins. However, our protein is not listed here, and we use this default part. This is one absorbance, one anticarotene. This is just a custom name. We'll just click it and it will show this baseline correction. These are some default options for this instrument and then we will click done. Now once we click done, the machine is showing clean both pedestals and load blank. That is, we have to clean this part. For cleaning we need scheme wide tissue papers, not normal tissue papers. The scheme wide tissue papers are specifically made for sensitive instruments. Now we will take this tissue paper, we'll gently lift up this handle and here you can see it's given the lower the arm to measure the blank. So before measuring the blank, we will just moist this tissue, we will use moist tissue paper and gently rub it out here. This is the pedestal where the sample is to be given out here and again this one. It is preferred not to directly pour water out here in this pedestal because sometimes there are sensitive parts or sensitive instrumental things maybe near there which might get affected. However, it is recommended to use a moist tissue paper in order to clean this. Now, once we have used this tissue paper to clean our pedestal, we will just use the blank. For the current part, we are using simple water as blank. So we are taking this 2 microliter pipette, and this is the tip. Now you are using pure water. You are using millicube water. To microliter millicube water. And gently at the tip, there is a tip here. We'll just give a drop, just single drop will do. And keep in mind and just check this one that there is no air bubble form out here. Now after that, what will happen is that we will just lower down this handle and this measurement will be carried out.
আরেকবার এটা বলো,
ঠিক আছে, এবার কোনটা কোনটা বলো বলো,
ওই লোয়ার ডাউন দ্য
হ্যান্ডেল ওটা বলতে হবে, এটা হয়ে যায়, তারপরে বলো যাবে।
হ্যাঁ, ও হয়তো মেজার করে নেবে, ব্ল্যাঙ্কিং দিয়ে গেছে তো।
অবশ্য ওটা ঠিক আছে, ওটা প্রথমে যখন করেছে ওই ক্লিপটা আছে, ওইটা এইখানে অ্যাড করে দেবো।
ঠিক আছে, ব্ল্যাঙ্কিং ইজ ডান, নাও ক্লিন অ্যান্ড
বলো। So we can see the blanking has been done automatically, and the option is coming that clean both pedestal and load sample. And one important thing is auto measurement is on. That is, if you want to make the measurement automatic, and if you want to make the measurements manually, we have to click a different option. However, it is convenient to use auto measurement. No harm in that. So again, we will just lift out this handle. Take this skim white tissue paper. Clean this one. No need to use moist tissue paper currently because it is already the bare solution. It is already the liquid in which the protein is dissolved. We will be taking a clean tip. Please note, don't use the previous tip. Sometimes you may use because the tip has been used in the solvent in which the protein is dissolved. But in biochemistry practical, it is better to use a clean tip each and every time when you are doing a new experiment. So here we have taken the protein solution dissolved in water currently. Again, we will be taking 2 microliter a protein sample. Carefully, we will be putting a drop. Given a drop, and now we will again lower down this handle carefully. See, now measuring, it's showing measuring data, automatically it's measuring the data. Here we can see this absorbance spectrum appears out here, and as we have previously seen in the UV spectra, there we can get an absorbance spectra. It is similar to that only. Here we can see this band around 280 which shows the protein absorbance spectra. Now one important thing to note from here is that the absorbance 280, out here we are showing absorbance 280.77. This is the absorbance from which we can calculate the concentration knowing the extinction coefficient at this wavelength. And now another important thing is absorbance 260 by 280. This value is 0.64, which clearly suggests that our protein solution is free from any sort of DNA or nucleic acid strand. This is actually used to measure the concentration of nucleic acid. The extent of nucleic acid present out here, DNA or So we have obtained this spectra. Here we can see this is the absorbance versus wavelength measurement. Here we can see the hump here we can see the hump which is for the protein absorbance around 280. From here we can see options are given here. A280 signifies the absorbance at 280 nanometer, and once we know the extinction coefficient of this protein at 280 nanometer, which is easily found in literature, here we can calculate the concentration from Lambert-Beer's law, which is absorbance equal to epsilon into concentration into length, and the path length here is standard is 1. Now here 260 and by 280 refers to whether any DNA or nucleic acid are present in the solution. The value 0.64 states that the protein solution is free from any sort of nucleic acid. So from here we can carry out our concentration measurement of this protein by using a very small amount of protein solution. Now we can end this experiment. We can either save the data or end the experiment. It's recommended to note down this absorbance value once the experiment is over. Now we will click this one, and here we are being shown an option where we can save this one.
Click this one.
It experiment click over.
Good.
Thank you.
Ready? Bolo.
Ready.
So what? So we can either save this data or we can note it down. Now we can end experiment. We can click this end experiment part. And once you click it, it is showing them whether you want to save this data. If we need, we can save it, but for the time being, once we need to estimate the concentration of protein, we just have to note down the absorbance value and no need to save this data. If you require, you should save it. Now, you have to end this experiment. Again, the option is showing, please clean the sample area. Now, how to clean this one? We have to again lift up this handle carefully. Take this clean wipe, moist it using distilled water, and clean the region, the pedestal where we have given the sample carefully. And once you have used this protein sample, kindly note to clean the pedestals so that it doesn't contaminate with the next sample which you or your lab mate might be using. After cleaning with water or with moist tissue paper, it is recommended to use a dry tissue paper or clean wipe to make the surface dry once again, free from water. And now we can lower down this handle. And if you wish to carry out any further experiment, we can click this one, touch to continue, and again we are directed to the home screen. So we can see how we can easily measure the concentration of any protein solution by using this nano UV. And unlike the previous UV experiment where we need around 200-300 microliter protein sample, we can measure the concentration easily by using a single drop. This generally requires for measurement of protein, generally a few proteins are there where you can't actually afford a huge concentration, a huge amount. Their proteins are quite expensive, and for that you need a small amount of protein. And now this instrument comes in that state where you can easily use the small amount of protein to measure the UV and find the concentration.